welcome everyone. I am Prashant and I am doing my M.Pharm in Pharmacology from Government College of Pharmacy, Bengaluru. This presentation was prepared under the guidance of our lecturer, Dr. Rajesh M.S., who is the Assistant Professor in Department of Pharmacology from the Government College of Pharmacy, Bengaluru. So in my last video, I explained about the topic which is genome organization, gene expression and its regulation. And in this video, I will be discussing about the topic which is basic principles of recombinant DNA technology and its application. So molecular biotechnology, it is an exciting scientific technique which is used to transfer specific units of genetic information from one organism to another. In this, conveyance of a gene relies on the techniques of genetic engineering. The main objective of this recombinant DNA technology is to often to create a useful product or a commercial process. The field of modern biotechnology started when recombinant human insulin, which is also called as humulin, produced by the bacteria, was first marketed in the United States in the year 1982. The effort leading up to this landmark event began in the early 1970s when research scientists developed protocols to construct new types of bacterial plasmids or vectors, which is prepared by cutting out and pasting pieces of DNA together to create a new piece of DNA that could be inserted into the host bacterium such as E. coli. And one of the most important technique under the molecular biotechnology is recombinant DNA technology. It is simply defined as the art of cutting and pasting of genes. And the first recombinant DNA molecule was synthesized in the year 1973 by a group of the scientists were named Berg, Boyer, Change and Stanley Cohen. And this technique uses a number of tools to construct a new combinations of DNA in a laboratory for different purposes. The recombinant DNA molecule, which is then constructed, is then introduced into an appropriate host cell, where it can be multiplied and generate many number of copies. This forms the basic concept of the process which is known as gene cloning. And gene cloning can generate unlimited number of DNA molecule by replication in a host cell. And the main principle of recombinant DNA technology is that DNA or a foreign DNA, or also called as insert DNA, is isolated from a donor organism. It is enzymatically cleaved at a specific site and it is joined with another DNA entity, which is also called as cloning vector, to form a new recombinant DNA molecule. This recombinant DNA molecule is then transferred into an host cell and it is maintained within it. And this process is known as transformation. This host cell then take up the DNA molecule, that recombinant DNA molecule, and it is identified and it is selected from those which are not taken up the DNA molecule. If required, the DNA construct can be created so that the protein product encoded by the cloned DNA sequence is produced in the host cell. The generation of recombinant DNA molecules is not a simple process. It requires a number of tools for the process of making the recombinant DNA technology. One such tool is known as the enzymes. The process of making a recombinant DNA requires a precise cutting and stitching of DNA molecules. And this enzymes helps in the process of cutting and modifying the DNA and finally resulting in the recombinant DNA molecule. The major enzymes which are required for the making of this DNA molecule are type 2 restriction endonucleases, DNA ligase, and alkaline phosphatase. This restriction endonucleases was first isolated by a group of 3 scientists, Smith, Wilcox and Kelly, in the year 1968. And the discovery of these enzymes marked the beginning of the recombinant DNA research and specific modification of DNA molecules. Since these restriction endonucleases are responsible for converting the DNA molecules into a specific number of fragments by cutting the molecule at certain specific sequences known as restriction sites. Hence, these enzymes are known as molecular scissors. These enzymes are formally designated as type 2 restriction endonucleases even though there are different types of endonucleases such as type 1, type 2, type 3 and even type 4. This type 2 restriction endonucleases are most commonly used, and it is known as restriction endonucleases or simply restriction enzymes. And one of the first type 2 restriction endonucleases to be isolated was from the bacteria Escherichia coli, and it was originally designated as E. coli. And this E. coli, it is a homodimeric protein that binds to a DNA region with specific palindromic sequence. The E. coli recognition sequence consists of 6 base pairs, and it is cut between the guanine and adenine residues on each strand. The nomenclature for this restriction enzymes consists of 3 parts or 4 parts, and abbreviation of the genus and the species of organism to 3 letters. For example, in Escherichia coli, it is known as E. coli 1, and the E represents the name of the genus, and the next 2 letters consist of a species, that is the coli, and the 2 words of coli is taken into the name, and it is then followed by a letter, number or combination of both of them to signify the strain of the species, and a Roman numeral letter to indicate the order in which the different restriction modification systems were found in the same organism or strain. Here we can see that the E. coli enzymes cleaves the internucleated bond between the oxygen of 3' carbon atom of the sugar of 1 nucleotide and the phosphate group attached to the 5' carbon atom of the adjacent nucleotide. The symmetrical staggered cleavage of DNA by E. coli produces 2 single-stranded complementary cut ends, each with extensions of 4 nucleotides known as sticky ends. In this case, each single-strand extension terminates with the 5' phosphate group, and the 3' hydroxyl group of the opposite strand is recessed. And along with E. coli enzyme, there are many other types of restriction enzymes such as IND3 digest and BAM H1 PST1 and E. coli 5th enzyme. Among this, the E. coli 5th enzyme produces the blunt ends, that is, it doesn't produce any elongating strands on one strand, and most of the enzymes produce sticky ends are also called as protruding ends, so that it will get stick to the another vector DNA molecule. And another type of enzymes which is required for this recombinant DNA technology is DNA ligase. This enzyme is responsible for the formation of phosphodiesterase linkage between 2 adjacent nucleotides, and thus joins 2 double-stranded DNA fragments. The phosphodiesterase linkage is formed between 3' OH group and 5' phosphate group of the pentose sugar of adjacent nucleotides of 2 different DNA fragments. In recombinant DNA experiments, DNA ligase is used to join different DNA fragments that are annealed by the sticky ends. There are different types of ligase enzymes from different sources, but the most frequently used one is T4 DNA ligase produced by T4 bacteriophage. And the next enzyme is alkaline phosphatase. In recombinant DNA technology, there is always a problem of self-ligation and the reformation of the original plasmid or the vector. This reduces the efficiency of the recombinant DNA formation. And this self-ligation is the process of annealing the sticky ends of the linearized vector without inserting the foreign DNA fragment. That is, the the cleaved molecule gets self-ligated to each other without any other foreign DNA fragment. On ligation, it will result in the original vector without any insert DNA, and this self-ligation can be prevented by using alkaline phosphatase. And the presence of the 3' OH group and the 5' phosphate group is the main reason for the ligase enzyme to act. If any of this group is absent, the DNA ligase cannot function. So this alkaline phosphatase acts by the removal of the phosphate group from the 5' end of the DNA molecule, resulting in the 5' OH group. Now the only chance of ligation is between the vector and the foreign DNA, resulting in the recombinant DNA molecule, as the insert DNA has 5' phosphate group. Thus, the self-ligation can be prevented and the efficiency of the recombinant DNA formation can then be increased. This enzyme is isolated by from some bacteria, which is called as bacterial alkaline phosphatase, or may be also isolated from the calf intestine alkaline phosphatase, and it is abbreviated as CAP. And in addition to this, there are many other types of enzymes which are used in minor applications, and one such is DNA polymerase. This DNA polymerase fills the gaps in duplexes by stepwise addition of nucleotides to 3' ends. And the next one is reverse transcriptase. This reverse transcriptase make a DNA copy of an RNA molecule. And next one is polynucleotide kinase. This adds a phosphate to the 5' OH and end of a polynucleotide to label it or permit ligation. And terminal transferase adds homopolymer trace to the 3' OH ends of the linear duplexes. And exonuclease 3 removes nucleotide residues from the 3' ends of a DNA strand. Recombinant DNA molecule is synthesized by various enzymatic process. Now it should be transferred into the host cell, and this transfer can be done with the help of vectors, which is the vehicle for cloning. This vectors acts as a vehicle for carrying the foreign DNA or recombinant DNA into the host cell, and whereby they go for multiplication. Usually small circular DNA molecules of bacterial origin are used as cloning vectors. And the DNA molecule should possess the following essential characters to act as a cloning vector. And the main characters, these are the following characters, that are origin of replication, selectable markers, multiple cloning sites or polylinker, and it should be small size. And origin of replication, it is required for autonomous replication of a plasmid using the host cell's replication machinery. Almost all commonly used plasmids are based on cold E1 origin of replication, and which is designated as ORI. Naturally occurring origin of replication are negatively regulated to keep the copy number down to reduce the load on the host cell replication machinery. With high copy number is disadvantages in a natural system. It is a desirable feature in a cloning vector, since the whole idea of the gene cloning is to easily isolate substantial quantities of a particular DNA sequence. And it should also consist of a selectable markers, which means when the organisms take up the recombinant DNA molecule, it should be identified by selections. And this selection can be done with the help of the markers, also known as selectable markers. And this selection can be divided into two types, that is positive selection and negative selection. Positive selection is used to identify bacteria that contains plasmid. The most common markers used for positive selection are antibiotic resistance gene, which are carried by the original R factors. While many antibiotics and resistance genes are available, the most commonly used resistance gene fall into two general classes, which are antibiotics affecting the cell wall synthesis and antibiotics affecting translation. And the antibiotics which are affecting the cell wall synthesis are ampicillin, or also beta-lactam antibiotics, whereas the antibiotics affecting the translation are tetracyclines or kanamycin or chloramphenicol. And the second type of selection is negative selection, and this selection system is necessary to distinguish between plasmids that are merely recircularized from those that carry a foreign DNA insert. It is used in order to identify those plasmids carrying a foreign DNA fragment. The site of insertion is chosen such that the insertion disrupts the selectable marker, and this phenomena is known as insertional activation. And the next character for a vector is, but it should contain a polylinker or multiple cloning sites. The vector should have a site specific for cloning the foreign DNA fragment, provided with one restriction site for most of the commonly used restriction endonucleases. All these unique restriction sites are grouped together in a small region of a vector known as multiple cloning sites. And the next character is small size. Relatively small vectors are more desirable because they increase the transformation efficiency, and are easy to manipulate. Small size also adds purification procedures to obtain intact plasmids. And this recombinant DNA technology, the vectors used for this recombinant DNA technology are mostly divided into six categories. They are known as plasmid cloning vectors, bacteriophage vectors, cosmetic cloning vectors, yeast artificial chromosomes, bacterial artificial chromosomes, and animal and plant vectors, which is also known as shuttle vectors. So now we will study each individual type of vectors in a very brief manner. The first one is plasmid cloning vectors. These are the vectors which are derived from the bacterial plasmids and are the most widely used. Behind it is because it is easily manipulated. Code E1 of a E. coli is an example of natural occurring plasmid. The origin of replication in almost all plasmid vectors is that of code E1. And the main examples for the plasmid cloning vectors are PBR322 vectors and PUC series vectors. And the PBR322 vectors is the most widely used, and some of the most important features is origin of replication, and the size of that PBR320 molecule, it is basically 4.4 kilobase pairs, and this is the most important because transformation efficiency is inversely proportional to the size, and about 10 kilobase pairs is the efficacy is very low. And another important feature of this PBR322 vectors is the copy number, and relatively high copy number which can be increased to 200-fold by treatment with a protein synthesis inhibitor such as chloramphenicol amplification. And it consists of two types of selectable markers, that carries two antibiotic resistance genes, which is ampicillin and tetracycline, which is designated as AMPR and TETR, and it consists of a cloning site. Some of these cloning sites are located in one of the antibiotic resistance gene, such as PST1, PVU1, and SAC1, and cloning into one of these sites inactivates the gene, allowing recombinants to be differentiated from non-recombinant known as insertional activation. And another type of plasmid cloning vectors are PUC series vectors, and it has three more important additional features compared to PBR322, and those are high copy number. The mutation within the origin of replication produces 500 to 600 copies of the plasmid per cell without amplification. And the most important techniques for this PUC series is the blue-white screening. This is the special form of insertional activation that can be used during the primary selection of the recombinant transformant rather than required a second round of screening. And it consists of a synthetic polylinker, and this is a piece of man-made DNA that contains several unique restriction sites. It has been inserted within the portion of the vector in such a way that it doesn't affect its expression. And this size of this PUC series vectors is about 2.7 kilobase pairs. And the next type of vectors is plasmid bacteriophage cloning vectors. These are derived from the viruses that specifically infect bacteria. Such viruses usually contain a comparatively small DNA genome surrounded by a protein coat. The viruses that infect bacteria are called bacteriophages. These bacteriophages, they infect bacterial cells by injecting their DNA into the host cytoplasm. This DNA then replicates and expresses the proteins required for the assembly of the new phage particles into the host cell. This process results in the production of large number of bacteriophage DNA molecules, which break the cells with a process known as lytic cycle. These new viral particles will reinfect the neighboring cells and the cycle continues. The natural way of transferring the viral DNA into a specific bacterial host has attracted scientists, and they have modified this viral genome DNA to use as a vectors for gene cloning. And this bacteriophage cloning vectors is again classified into two types, such as phage cloning vectors and M13 cloning vectors. These phage cloning vectors, these are the engineered version of the alpha bacteriophage that infects E. coli. This vector has a double-stranded linear DNA genome having a size of around 48.5 kilobase pairs, are about 12 bases at the ends of this DNA molecule are unpaired and complementary and therefore are sticky or cohesive and also known as cos sites. The lambda phage vector is designed in such a way that the central region of the linear chromosome is cut with a restriction enzyme and is replaced with the foreign DNA digested with the same restriction enzyme. And these are then packaged and assembled into the bacterial viral protein, which is surrounded by a protein coat, and this is then ingested into the bacteria such as E. coli. And the next type of bacterial cloning vectors are M13 based cloning vectors. These are the vectors of the family derived from the bacteriophage M13. This is a male-specific lysogenic filamentous phage with a circular single-stranded DNA genome of about 6.4 kilobase pairs in length. On infection, this molecule is transferred to E. coli and converted into a double-stranded replicative form. The replication continues, and when there are more than 100 copies of DNA in the cell, the DNA replications become asymmetric and produce copies of the original single-stranded DNA molecule, which are packaged into infective particles and extruded from the cell. The host is never lysed, but the cell continues to grow throughout the infection, although at a significantly reduced rate. This difference in the growth rate between infected and the uninfected cell produce characteristic flakes when M13 phages are plated on a suitable host. And the main advantage of this M13 based vectors are that they contain the same polylinker and alpha peptide fragments as the PUC series, and the recombinant can be selected by blue-white color test. And since the size of this genome is very low, it is very easy to handle. And the next type of cloning vectors are cosmate cloning vectors. These were among the first large insert cloning vehicles which are developed. A cosmate can be defined as a plasmid that contains a cos site from the lambda phage genome. The simplest cosmic vector has cold even origin of replication, selectable markers such as AMPR or tetracyclines, and suitable polylinker site and lambda cos site. And ligation of the cosmate vector and foreign DNA fragments of sizes up to 45 kilobase pairs is similar to ligation into a lambda substitution vector. And the next one is yeast artificial chromosomes. These are the vectors that enable the formation of artificial chromosomes with foreign DNA fragments and cloning into yeast. These are used for the cloning of very large DNA fragments in the range of about 500 to 1000 kilobase pairs. These vectors are constructed with the yeast telomere sequences at each end, yeast centromere sequence, and yeast autonomously replicating sequences for replication. In addition to this, there are restriction sites for the DNA insertion and selectable markers such as amino acid dependence and antibiotic resistance on each arm. The recombinant DNA is constructed by ligating high molecular weight to the YSC arms using the appropriate restriction enzymes. These recombinant vectors or the artificial chromosomes are used for the transformation of yeast cells by electroporation, and the transformed cells are selected on suitable growth medium. Bacterial artificial chromosomes are also known as BSEs. These are the cloning vectors based on the extra chromosomal plasmids of E. coli called F factor or fertility factor. These vectors enable the construction of artificial chromosomes which can be cloned in E. coli. This vector is useful for cloning DNA fragments of up to 350 kilobase pairs, but can be handled like regular bacterial plasmid vectors, and it is very useful for sequencing large stretches of chromosomal DNA. Like any other vector, this BSE also contain ORA sequence derived from E. coli plasmid effector. Modified cloning seeds are also known as pol linker, and these are having a unique restriction sites and suitable selectable markers. Both YSC and also BSCs are very useful in genome sequencing projects such as Human Genome Project. And then next one is shuttle vectors. These are some vectors developed for transforming plant and animal cells. Numerous vector systems have been developed for use in eukaryotic systems. The most common of these are often called as shuttle vectors as they replicate both in prokaryotic and eukaryotic host. In general, DNA manipulations and characterizations are done in prokaryotic systems, and then the manipulated DNA is reintroduced to eukaryotic systems for functional analysis. The common features of these shuttle vectors are the following. They are capable of replicating in two or more type of hosts including prokaryotic and eukaryotic, and they replicate autonomously or integrate into the host genome and replicate when the host cell multiplies. These vectors are commonly used for transporting genes from one organism to another, that is transforming animal and plant cells. And the one such shuttle vectors are mammalian vectors. These are the vectors developed for use in mammalian tissue culture. These eukaryotic origins of replication are typically derived from well-characterized mammalian virus such as simian virus 40 with SV40ORI and large antigen system or mononucleosis. At present, retrovirus-based vectors are the most commonly used vectors for cloning genes in mammalian cells. In addition to the origin of replication, these shuttle vectors also carry antibiotic resistance genes which function in the eukaryotic cells. And finally, artificial chromosome vectors have been developed for use in mammalian cells known as mammalian artificial chromosomes, also known as MACS. And next one is plant vectors. These shuttle vectors are based on certain viruses and bacteria which are pathogenic to plants. Plant viruses such as cauliflower mosaic virus or tobacco mosaic virus were used for developing vectors for plant cell transformation, but it has limited success. The most successful shuttle vectors developed for the plant system are those that are based on the type plasmid of Agrobacterium tumefaciens, a bacterium that causes tumor formation in plants. This has been successfully exploited for the development of a tumefaciens based vectors for the genetic engineering of the plants. The success in plant genetic engineering and the development of a number of transgenic plants is mainly due to the development of vectors based on type plasmid and A tumefaciens. So by this we end the various vectors which are used for the recombinant DNA technology. In my next video, I will be discussing about the host cells which are used and what are the techniques for the transformation of this recombinant DNA molecule into the host cells, and then we will study about the applications of recombinant DNA technology. Thank you.